Welcome, everybody. Good afternoon. This is the final lecture of the 2021 spring season for Frontiers of Brain Health. And we are dedicated at the Center for Brain Health to bringing you some of the most exciting, cutting-edge developments in brain research. I'm Dan Krozik, the Deputy Director at the Center for Brain Health, and also the Debbie and Jim Francis Chair in UT Dallas. We have dedicated ourselves for 3 decades to understanding cognitive neuroscience of the mind and focusing particularly on neuroplasticity and the tie-ins between the brain and our everyday lives. And today we have an extremely exciting lecture of one of the true innovators, a close colleague of ours, Ed Boyden. Ed goes way back with the center, actually. We were talking about the first time he gave a talk, and he was not yet a father. And now we just saw photos of his kids who are running chemistry experiments on the weekends. So we've come a long way. Ed develops new tools. He's an innovator and an engineer for analyzing, engineering, and observing neural circuits. His methods include synthetic biology, nanotechnology, chemistry, electrical engineering, and optics to reveal fundamental mechanisms about the brain. He was an early pioneer, actually the co-inventor of optogenetics, which has become one of the dominant technologies in behavioral neuroscience for understanding how we can drive neural circuits, and it offered investigators a whole new window into investigating the brain. Ed did that work in graduate school alongside his PhD. So from a very early point in his career, he's been a true innovator at Stanford, where he did his PhD. He's then been at MIT ever since. He led a team that invented expansion microscopy in recent years. This is a technique that allows researchers to expand nanoscale features of specimens through water absorption to a size where you can see those features using standard microscopes. And he is integrating these technologies to create detailed maps and models of brain circuitry, so really contributing to the fundamental understanding of the brain. So Ed, of course, has been at MIT for a number of years, where he is the Y. Eva Tan Professor in Neurotechnology and Professor of Brain and Cognitive Sciences, with joint appointments in the program in Media Arts and Sciences and the Department of Biological Engineering. Ed was selected to become a Howard Hughes Medical Institute investigator in 2018. He has been awarded the NIH Director's Transformative Research Award 3 times and received an NIH Director's Pioneer Award in 2013. He is a member of the National Academy of Sciences, a member of the American Academy of Arts and Sciences, a member of the American Institute for Medical and Biological Engineering, the College of Fellows. He is a great speaker. He has 2 TED Talks that have millions of views. In short, Ed is what we'd call a rock star in scientific terms. So without further ado, we will welcome Ed, and please use the Q&A function if you have a question, because we'll get to those at the end of the talk. Take it away, Ed.
Hello, everybody. It's really great to be here virtually. Sorry I cannot be there in person. Hopefully soon that will be possible. I direct a group at MIT that works on technologies for understanding and repairing the brain. We build these tools and send them to thousands of researchers all over the world and help them make discoveries. And then we often get involved, if something turns out to be interesting, to think about how this can translate to help people as well. And so today I'll tell you a bunch of examples about technologies we're building that are revealing things that we don't know about the brain, and how we're trying to take them and the insights that are derived from them into a translational path as well. Let me try some screen sharing here. So why is the brain so hard to understand and so hard to fix? We just saw, for example, the development of COVID vaccines in what might be record time, shortly after the genome was sequenced. Well, the problem with a lot of these situations related to the brain is the enormous spatial and temporal complexity. So from a space standpoint, brain cells are enormous things. They're, in the human brain, centimeters in spatial extent, by far the largest cells in the body. But the wiring of the brain is nanoscale, zillions of times smaller. And the biomolecules that are encoded by the genome, literally thousands of kinds of biomolecules, they too are nanoscale and often arranged with nanoscale precision. So how can you understand something that goes from nano to macro? But it's worse. The time axis is complicated. If you're studying how a child learns language or how the brain ages or the progression of Alzheimer's, list goes on and on. These are slow events. They take days, months, even years. But the fastest signals of the brain are very fast, 1,000th of a second in duration, which is very quick from a biological time standpoint. So I often say our group's mission is to help neuroscientists, biologists, clinicians to cross these spatial and temporal barriers. So today I'll tell you 3 short stories, each of which begins with an invention, and then we'll talk about what kinds of discoveries we can make with them and how those can lead to the design of things that can go potentially into the clinic. I'll tell you 1 story about space and 2 about time. Let's start with space. So how do you image nano objects like biomolecules across a large object like a brain? Well, many people have invented nanoimaging methods. Super resolution microscopy won a Nobel Prize. Electron microscopy has been used for decades to map nanoscale features. But all these methods struggle with large objects like brains. And also you need specialized equipment, which can be expensive and require training. So in our group, we often think about doing the opposite of the way things have been done. We started wondering, what if we try the opposite of the last 300 years? So for 300 years, people have used a lens to magnify an image of an object. What if we try to magnify the object itself? We call this expansion microscopy. And we thought of many ways to do it, but here's the way that worked the earliest and is now spreading very rapidly throughout biology. We take a preserved, not living, specimen, like a piece of brain tissue. We chemically weave a dense spiderweb-like mesh of swallowable polymer inside cells and outside cells, in between biomolecules and around biomolecules, very densely permeating. We anchor key biomolecules to the polymer, soften the specimen, add water, and the polymer, which is sort of like the stuff in baby diapers, will swell. And as it swells, it'll pull apart the biomolecules. So let's go through this a little bit more slowly. Here's the goal. We want to take a cell like the brain cell on the left and turn it to something like on the right, a constellation of biomolecules hovering in space. 2 biomolecules that we're touching are now some minimum distance apart. 2 biomolecules that are some distance apart will be scaled up by a linear factor. Now to make this work, we had to invent a bunch of chemistries. In this cartoon, biomolecules are shown in brown. We had to invent anchors, little handles that would bind to each biomolecule so we can pull them apart. And we now have anchors for DNA, RNA, proteins, and we're working on more. Next, we have to weave that dense spiderweb-like mesh of swallowable polymer. We use a method called free radical synthesis. The little white spheres are monomers, and they self-assemble into long chains, a strategy that was first used in 1981 in tissues. But here, we're using a swallowable polymer, and it connects to the handles. Next, we soften the specimen. We can use heat, detergent, enzymes, or all 3. And then we add water, and the baby diaper polymer, sodium polyacrylate, will absorb the water. As it absorbs the water, because of those handles I told you about, the biomolecules get pulled apart. So amazingly, this worked. By design, of course, we wanted this process to be very even, a very dense polymer. We soften the specimen, but it's always nice to see it with your eyes. Panel B is a piece of the mouse brain. Panel C, about a day and a half later, we blew it up by 100-fold in volume, about 4.5 times in each direction. The polymer starts out compact and it ends up extended. So we can now see nanofeatures on ordinary microscopes. This technique democratizes nanoimaging because now anybody can do nanoimaging. Physically magnify the object, and then you could use existing hardware to see nanofeatures. On the left, we're zooming in from top to bottom. The color code is shown on the left. These are fluorescent antibodies that target specific biomolecules. On the right are the same fields of view as the left, but after we expanded it. And the features now can be cleanly resolved. We can see synaptic connections between brain cells and measure the distance between the pre- and postsynaptic proteins. We get the same answer that Catherine Dulac and Jiawei Zhuang measured many years ago with STORM, a classical super resolution technique invented by Jiawei. But now we can use a regular old confocal microscope. This is work that initially was done by Fei Chen and Paul Tilburg when they were students in my group. We can label mitochondrial and lysosomal proteins, as you see in the upper right. We can label myelin and map it out. This is work we did with Eric Betzig's group, done by Reigao Shoasano and Gokul Padula. In this example, we went beyond using an ordinary microscope. It's nice that expansion democratizes nanoimaging, but it also makes an opportunity for specialized microscopes to do very fast imaging. And so we, in this paper, I think set what might still be the world speed record for nanoimaging, about 1,000 times faster than a classical method. You can put in artificial proteins and image their presence. Here we're putting fluorescent proteins derived from coral and jellyfish and so forth into brain cells. And we can then try to map the wiring of the brain. At the top middle, it of course is hard because the wiring of the brain is nanoscale. The upper right, though, is after we expand. And now we can see the finest wiring of the brain. Many groups are now doing some kind of color coding, followed by expansion to map brain circuits. Now, what I showed you in the past 2 slides is visualization of proteins, but we can also visualize other molecule types like nucleic acids. Here, in work that Ozwasi and Fei Chen and others did, we expanded proteins and RNAs away from each other, and then labeled RNAs with in situ hybridization. That's the purple dots. So now we can map the location of individual expressed genes with nanoprecision in intact brain tissue. Recently, we showed that you could do multiplexing too. After all, there's so many genes. Can we understand what they all do? We expand and then bring in a hybridization probe shown in purple, and it has a small barcode on it. We can then sequence that barcode. When you sequence, you just copy, right? You're copying bases, and as you do, you add fluorescent ones, and so each gene will blink out its identity. We can make libraries of dozens of such probes, each with a barcode, and then apply them to an expanded brain, sequence the barcode, and look at the cell types and properties. So working with the Allen Institute for Brain Science, we synthesized several dozen probes against cortical cell types. We did the hybridization and sequencing, and here are the clusters that emerged using a standard clustering method. The upper right shows the cells superimposed over the visual cortex. Here's layer 1 on the right, layer 6 on the left. The colors are the same as in the upper left, but now we can show you where those cells are. And because expansion gives you great resolution, we can look at the identity and location of individual expressed genes, even in nano compartments like synapses. Here we took some data sets from Erin Schumann's papers, where she's been looking at how gene expression occurs in the dendrites of neurons, and we can localize individual RNAs in these dendritic arbors. As a side note, collaborating with Jason Buenrostro's group and Fei Chen's group, led by Andrew Payne, Zach Chang, and Paul Reginato, we showed that we could also do in situ sequencing of the genome. Bring in little barcodes into the genome, sequence them, just like I told you, and now we can see not only the genetic code, but the shape of the genome. And they're fascinating patterns about how genomes are organized in cells. And now we can see them. Back to expansion. How big can you expand? Well, we can take a specimen, polymerize and expand it, form a second polymer and expand it again. When J.B. Chang was in the group, we prototyped this, and recently Deblina Sarkar, Jinyoung Kang, and Oswasi, with great help from Margaret Schroeder and Amuche Emanari and Christina Kitko, have developed important versions of this. We showed that we could do this double expansion. And it has a really cool added bonus. Ordinarily, when you visualize biomolecules, you label them. And in the upper right of this cartoon, you can see the red and green antibodies binding to the outside of a complex. But what about the inside proteins? How do we see them? Well, if we pull apart the biomolecules, now the blue and yellow antibodies can get in. That means that we might be able to convert invisible things into visible ones. This slide's a bit crowded, but let me walk you through it. In panels B, C, and D, we stained with fluorescent antibodies against biomolecules. Yellow is pre-expansion, not much yellow, and purple is post-expansion. When we pulled the molecules apart, we could label them. Not all proteins were thus crowded. Here in F, G, and H, you can see some molecules where the pre and post-expansion are quite similar. Using this technique, we can map out the nanostructures that make neurons communicate. Now that we can look at, for example, a calcium channel that we could not see before, we asked whether it has nanostructures that coordinate the precision of neural signaling. And it does. We see not uniform distributions of proteins, but highly precisely coordinated organizations of these molecules that let them, we think, preferentially talk to each other. This is work we're doing with Tom Blanpe's group. With Li Wei Sai, we started wondering, could we investigate Alzheimer's disease at a deeper level? Amyloid plaques and other protein aggregates are very densely packed. If we expand them, can we label them better and see them? Well, starting with a 5x FAD mouse, and now we're starting to explore human tissues, we did pre-expansion labeling followed by post-expansion labeling, again in yellow and purple. I hope you agree there's a lot more purple than there is yellow. And indeed, we even saw new structures which we don't think anybody's seen before. These almost periodic amyloid dots. We saw this with multiple different antibodies, and we never saw them in wild-type mice. Could it be that in neurodegeneration, we are looking at things with older tools so that we don't see everything we need to see? Hopefully, this expansion method will allow us to get down to the ground truth, nanomapping of the building blocks of life and how they go wrong. By the way, you can apply this technique to other tissues than the brain. When Yongxin Zhao and Octavia Bucure worked with me and with several pathologists, Astrid Wines, Andy Beck, and others, we showed we could expand to human tissues. Normal on the left, cancer containing on the right, from top to bottom, prostate, lung, breast, pancreas. Color coded in the upper right. And we showed that we could actually analyze tumors earlier than if you don't expand. After all, early in a disease, the changes are small. But if we make those small changes big, maybe we can do better diagnosis. Something like 7 out of the 10 leading causes of death, if you detect them earlier, you could save more lives. Well, can we detect earlier diseases by magnifying those small changes? So in this paper, we showed that that was possible with a breast cancer study using breast cancer biopsies from actual human patients. And our hope is that we can continue to advance early diagnostics in the time to come. So to summarize this first part of the talk, we've discovered that we can physically magnify objects. This allows us to do nanoimaging with regular hardware. And that's important because biology and medicine, I think, are fundamentally nanoscale sciences, right? Biomolecules are nanoscale. They're organized with nanoscale precision. The only reason we don't really do nanoimaging routinely is it's hard. But now we've made it easy. Expansion is in use by hundreds of groups. The 250th experiment was just published. It's being applied to the human kidney, to the microbiome, to stress granules, to the fruit fly brain, and everything in between. Our website, expansionmicroscopy.org, has protocols on how to do it, including really detailed photographic step-by-step tutorials, including little things like how do you handle a cover slip? How do you pick up a hydrogel with a paintbrush? So if we can help in any way, we're excited that we've democratized nanoimaging. We'd love to help understand brain diseases and reveal new targets. And already we're working to build a network to advance early disease detection. Since again, detecting diseases earlier could help save a lot of lives. Okay, that ends the story on space. And now let's transition to time, because of course, expansion doesn't work on living things. How can we control and observe high-speed living dynamics? Well, let's start with optogenetics. So in optogenetics, opto for light, and genetics because, as you'll see, we use genetically encoded things. This started when Carl Dijsselhoff and I were both students at Stanford, and we wanted a way to control the brain. And so we just started going through all the laws of physics. There's only so many kinds of energy you can deliver to the brain. Electric fields, magnetic fields, light, sound, a few other things. We thought light would be the best if you could get away with it because it's fast and precise. You could bring light into the brain the same way that electrodes are brought into the brain. But how do you make brain cells sense light? I'm just going to take a sip of tea here. Well, I became very fascinated by a class of molecule known as microbial opsins. These are membrane proteins, like in the lower left, that have a vitamin A relative that senses light, and they sit in the membranes of microbes that live in salty water. The first to be found a half century ago was a light-driven proton pump. A decade later, several groups found light-driven chloride pumps. These pumps convert sunlight into chemical energy. And then around the turn of the millennium, people found light-driven ion channels. Now, to make a long story short, what we found over the years is that members of all three of these molecule classes can be found that are safe, fast, and effective for activating or quieting neurons. So by activating a neuron, you can figure out what it does, what is sufficient to trigger. By silencing neurons, you can figure out what they're needed for. So Brian Chow, Xue Han, Amy Chuang, when they worked with me, we found certain light-driven proton pumps or chloride pumps could be put into neurons, shine light of the right color, like green or yellow, and you could turn the neurons off. For Amy's thesis, we found that red light could drive a molecule called JAWS to turn off neurons. Red light goes deeper in the body than other colors of visible light. And we can also activate neurons. Carl and I found that one of the light-driven ion channels expressed in neurons would let you activate it with blue light. And Nathan Klepecki, when he was a student in my group, found that we could make a red light version as well and activate the neurons with red light. And then others like Or Shemesh, collaborating between my group and Valentina Emiliani's group, have tried to make the technology very precise. So these tools are in use by literally thousands of groups, thousands of researchers anyway, to activate and silence neural activity. And I just want to give you an example. And I picked this example partly because I'm involved with it, but partly because it exemplifies the same sort of paradigm that I mentioned earlier. Build a tool, give it to everybody. As people use it, they'll discover new scientific insights. And then, can you design a practical version that can go out into the world? Much like how expansion was an invention, and then we wanted to map the brain, but pathologists wanted us to look at cancer, and now maybe it could help with early disease diagnosis. So Li Wei Sai led a project, Henna Yakarina and Annabelle Singer, working between her group and my group, are the co-first authors of this paper. And what they did was to use optogenetics to drive the brain of Alzheimer's model mice at a certain frequency, 40 times a second. And amazingly, the mice got better. Amyloid and tau got cleaned up, microglia became less inflammatory, blood flow increased, cognition improved. Who would have thought that a brainwave might help address one of the most intractable brain diseases of all time? Well, under Liwei's leadership, the teams went on to find out, okay, got the invention, the optogenetics, got the discovery, maybe a brainwave could help clean up Alzheimer's. The next step is, could you design a practical way of doing it? One that doesn't require gene therapy in the brain. And so under Liwei's leadership, the teams went on to show that you could induce the same brainwave by having no optogenetics needed, by having the mice watch a flickering light or hear a clicking sound. Basically, to have the mice watch a movie. And now there are clinical trials with actual Alzheimer's patients seeing if movies can help treat Alzheimer's. And so it's very early days. As everybody knows, it was hard to do clinical trials in the current era. But there are now a couple abstracts and preprints floating about suggesting that there might be positive effects in human patients. Inspired by this idea of invent, discover, design, our own group has been also working on less invasive ways to control neurons. When Nir Grossman was in my group, we asked, could we use the fact that brain cells are nonlinear to focus the effects of electricity? Put some high frequencies outside the brain, and where they overlap, you would get a difference frequency. If the high frequencies are fast enough, the brain won't keep up. But if the difference frequency is low enough, the neurons will fire. And we found that was true. We could do temporal interference, apply 2 high frequencies to the brain, and we found neurons would fire at the difference. So potentially, we could use this as a more general way of focusing the effects of neural stimulation deep in the brain. So to summarize the second short story, we discovered that we can use natural molecules to make brain cells sense light. It's being used by lots of people to discover new patterns of brain activity that could have potential disease-related or health-promoting effects. And our hope is to also help people design practical implementations, non-invasive, ideally, that allow the healing of diseases. Now, to transition into the third part of the talk, I think it's important to acknowledge with optogenetics just how lucky we were. These molecules from microbes, just out of the box, no modification needed, were fast enough, safe enough, and strong enough to mediate the control of brain activity. What about the opposite? Could you watch the brain in action? Well, there we were not so lucky. The natural world has not made molecules that let us eavesdrop on what the brain is doing all the time. So we thought, if nature won't evolve those molecules, let's do the evolution in the lab. When Erica Jung and Kirill Piatkovich were postdocs in my group, we built a robotic platform to evolve new molecular tools. How does it work? Take a gene and make millions of mutants. For any goal, some are better and some are worse. Some of the mutants are better and some of the mutants are worse. Put them into a million cells in a dish and use a robotic arm to pick out the cells and therefore the mutants that are better for your goal. We wondered if we could make a fluorescent voltage indicator, the opposite of optogenetics, if you will. We began with the molecule Quasar 2 from Adam Cohen's group at Harvard. And to make a long story short, in 2 rounds of evolution, we made the molecule better expressing, brighter, and photostable. And indeed, collaborating with Shahan's group, we were able to show that we could image the voltage of neurons in awake-behaving mice with a simple microscope. So it's appealing to think, what if we can make an all-optical interface to the brain? We could use it to study the brain and inspire new scientific discoveries that could lead to new designs. But someday, maybe this could be a brain interface in its own right. It's non-contact. You could image the activity without having to touch the cells. And here's an example where we're looking at many cells at once in the brain of an awake mouse. So we're very excited to build all sorts of molecular tools using our robotic method. But we're not the first to try to make molecular tools to look at cellular signals. There are hundreds out there. These are screenshots from Xin Zhang's database from her lab at UCSD. And it lists hundreds of fluorescent sensors of different cellular signals. Wouldn't it be great if we could understand how all those signals talk to each other? Well, to do that, you need to look at many signals at once. That's hard. There's so many important signals: calcium, messengers like cyclic AMP, kinases like protein kinase A. The human genome contains tens of thousands of genes that might be active in the body at any time. How can you see all that? Well, until recently, the answer was make molecules different colors. One could be green and one could be red, and then you could look at both at once. But it's hard to make new molecules that are indicators. Also, what if you want to do 3 things, or 4, or 5? Well, Shannon Johnson and Qingyang Gu and my group, we decided to try a very different way of mapping signals. What if you could put reporters of one kind at the points labeled 1 within a cell? And what if you could put the reporters of a second signal at the points labeled 2? While the cell's alive, each point will blink out a little movie of the signal that it reports. When the experiment's done, you can come in and label those points and figure out which signal was at each point. And here's how we made it work. We can take different off-the-shelf fluorescent reporters and fuse them to different self-assembling peptides. That will help them cluster at distinct stationary points in space. If each point also has an epitope, we can label it with an antibody and figure out which kind of reporter was at each location. Here's a little animation explaining the concept. The live cell image, you see a lot of green dots. And then after the experiment's done, you can label those dots and figure out which reporter was at each dot. Does it work? Well, panel B shows a much loved calcium indicator, GCAMP6, in a cultured neuron. Panel C, the same calcium indicator fused to a self-assembling peptide set. And look, it clustered. We did lots of control experiments looking at signal to noise, kinetics, physiology, cell death, cell health, synapse density, microglial inflammation, and the list goes on and on and on. And we never saw any differences in cell health or physiology between the clustered indicator and the regular one. So we can take a bunch of indicators. Here we took 3 off-the-shelf indicators, fused them to different self-assembling peptides and to different epitopes. The green images are the live images. The false color images are when the staining was done at the end of the experiment. And look, they end up at different points, and we can figure out which signal is at each point. So now, for the first time, we can take existing indicators and use them to map out how signals occur in cells. Here we're looking at calcium, cyclic AMP, and pKa, some of the most important molecules in learning and memory and all sorts of other fields in biology. We drive forskolin, which makes cyclic AMP, and then we can image all 3 signals. And we can derive relationships between those signals. In H, for example, some neurons had slow calcium. In I, some neurons had fast calcium. Turns out in panel L, we can predict the protein kinase A response from the calcium. Fast calcium neurons had strong protein kinase A. Slow calcium neurons had weak protein kinase A. So we can now derive relationships between different signals in a living cell. We can do this in tissue from the living mouse brain. Here we express these in the hippocampus of the living mouse brain. We cut acute slices to facilitate imaging. But we saw the same relationship. Fast calcium neurons had strong pKa, and slow calcium neurons had weak pKa. We can do 4 signals at once. Here we threw in a protein kinase C indicator. Again, green is live. False color is the labeling to figure out which signal is at each point. And we can monitor 4 signals at once, or even 5 signals at once. Here we threw in an ERK kinase indicator. So there's so many signals that we need to understand in order to really pinpoint what goes on in healthy and disease states. And so to end the third part of the technology part of the talk, we were not lucky in the sense that we were with optogenetics, where the natural world just gave us stuff. But we kind of decided to become lucky by building robots to optimize our luck and by using existing tools in clever ways. Before I end, I want to talk a little bit about integration. The old model, of course, is we build a tool, we share it with lots of people, we help them make discoveries, and those discoveries can lead to designs that can have clinical impact. But could we integrate these tools into a workflow that could yield really novel computational models or other insights into how entire brains work? What if we could image the signals going from bottom to top here and perturb them, and then at the end of the day, make a map of the molecules and the wiring? Could we make a computational model of the brain? Could you simulate a decision or an emotion? In recent years, we've started to think about how to do this. One idea is to start with small brains, like fish and worms. One of the great lessons of biology is start with something small and try to solve it completely. The first genome to be sequenced was not the human genome, but a small virus, and so on and so forth. When Limor Freifel was in our group, she showed that we could expand the larval zebrafish brain with 100,000 neurons. J.U. showed that we could expand the worm C. elegans with 302 neurons. So what if we could make a map of molecules throughout cells and cells throughout networks? Would that be enough potentially to go in this direction of brain simulation? And we can also do imaging of signals in the living fish, as you can see here, where we're doing voltage imaging, or in the living worm, where again, we're doing voltage imaging. So stay tuned. We hope to see if we can make some advances in this front in the next 5 years or so. In the meantime, we share all these tools freely. Our website, synthneuro.org, in the upper right, has tutorials and ways of getting these reagents. And if I can help in any way, email me. We always love to figure out how we can help people solve problems. And with that, I'd be happy to take questions.
Okay, Ed, thank you. Fantastic glimpse into the future, it seems like, with all this potential. So many different avenues we could probe for questions. I will start us off with something that was on my mind regarding the expansion microscopy work that you're doing. I was thinking like dermatology would be a really obvious sort of application space for this, because the skin it doesn't require you to go internally with, as much as you would with other types of tumors. But maybe more broadly, the better question is sort of what domain do you see most actionable or the most potential in for that expansion work?
Well, it's being applied to literally hundreds of problems. Biology is about nanoscale building blocks and how they interact in the end of the day. And it's being applied to look at gut microbes. It's being applied to look at nephrotic kidney diseases. It's being applied to all manner of cancer biology. Recently, we were part of a collaboration that showed that maybe we can map out tumor immune interactions in actual human cancer biopsies. A couple of years ago, the Center for Disease Control gave their top science prize to a CDC team that used expansion to try to figure out how viruses spread from cell to cell, a very germane topic for the public health right now. People are trying to apply it to look at, it's been applied to epileptic brain tissue removed from the operating room to try to figure out at the nanoscale what's happening between the epileptic brain and the blood vessels. And a group used expansion to pinpoint some molecular changes that had not been seen before. It's being applied to all sorts of species I've never heard of. Yeah, the list goes on and on. So it's hard to pick something specific just because I really do think biology is fundamentally about nanoscale building blocks and how they interact. And so now if we can empower everybody to think about the building blocks of their system, because it's no longer a expensive thing that takes years and zillions of dollars to do, that could be great.
Fascinating. Yeah, there's almost too many potential directions one might go there. Here's another question from the audience, and one that I was curious about as well, regarding that frequency movie for the potential for Alzheimer's. Just the attendee is curious how close might that be in terms of being a treatment. You mentioned a clinical trial. Wonder just what are your thoughts on the clinical potential for that?
Yeah, so there's one paper already on a feasibility trial of the gamma flicker from Annabelle Singer's group. I can usually I can paste things into the chat window. I wonder if that's possible. I don't see a chat window though. Oh well. Yeah, look up Annabelle Singer on Google Scholar, and she just had a paper that came out on May 13th, where they show that with preliminary effects that this flickering movie helped change immune factors in the human nervous system. And then out of MIT and Harvard, Lee Wei's group led a study, which is it's in preprint form on medRxiv, MEDRxiv, which has a preliminary clinical trial of the gamma on Alzheimer's. And this also showed improvements in cognition and decreased brain atrophy. Yeah, so it's getting quite exciting. These are all small scale trials, and so we should be cautious about anything. Lots of Alzheimer's trials look tantalizing, and then the big trial fails. But we're bravely plunging on.
Thank you. Here's a timely question. Can expansion microscopy help us with early detection of mutations of virus, particularly things like COVID-19?
Yeah, a bunch of people have approached me, and I think some groups are already applying expansion to study effects of COVID-19. As I mentioned, I mentioned ways of doing multiplex RNA and DNA analysis in the first half of the talk, and a lot of people are jumping on that as well. So yes, all of the above.
I think we have the chat disabled just so that we could channel the questions to the Q&A. Some people are kind of wondering about the chat function, so that's not possible. No, but definitely look up those references that Ed mentioned. Another question regarding the driving neurons with light. It's a brilliant innovation that has really taken off. I wonder if you could comment on the applications you see for photobiotic modulation of the brain. This is a technique that's emerging, and that just seems to align so well with this more micro level work. I wonder if you're familiar with some of the cognitive trials using transcranial photobiotic modulation, and if your work maybe better informs them or could possibly better inform them.
I've heard about these, but it's certainly not my area of expertise. I guess they're shining light on the normal brain, and the brain has something that is absorbing the light and changing. In our case, we're adding a factor from bacteria or algae or a fungus into the brain. So it's almost like installing a little solar panel there, and then we shine light on it. So it's a very different technology. Although both use light, but they don't have anything in common to my knowledge.
Okay. A couple more disorders questions in the queue here. What are your thoughts on the potential for any Parkinson's disease related clinical trials deriving from your work, or multiple sclerosis as well?
Yeah. a lot of people are applying the different tools to all the above. I mean, a preprint just came out, although I haven't had time to read it, where they're using expansion to look at the aggregates found in Parkinson's disease. So again, I haven't had time to read it yet. There's just so much going on with using the tools. People have applied optogenetics to try to discover sites in the brain that might be good for modulation for Parkinson's. There are several papers on that. So that's very exciting. Yeah, I know less about multiple sclerosis. So I'm trying to think off the top of my head if I've read a paper on that, but yeah, if somebody who's an expert on MS would like us to help with our tools, that's what we do. We help people use our technologies. But yeah, Parkinson's, I've seen several papers now where expansion microscopy and optogenetics are being applied to find better targets.
Here's a sort of micro level question on the expansion sample preps. Do you have a loss of biomolecules? Or I guess maybe more broadly, are there downsides to undergoing that expansion process that you're concerned about in terms of its limitations?
So remember I said there are little handles or anchors that we attach to biomolecules? That's what ties them to the polymer. If they're not anchored, they will be lost. And so, but we can anchor the major biomolecule classes that 99% of, I'm just making that number up, but some vast fraction of the community is trying to image: DNA, RNA, proteins, and we are working on ways to look at lipids and other molecules as well. So it seems to meet what people's needs are. I mean, no imaging method is guaranteed to keep all the molecules there. Many molecules will start to fall apart the instant that you preserve a specimen, for example. In terms of downsides, that there is one major downside, which is you can't do expansion on a living thing. Yeah, I just haven't figured out how to do that, and it might not be possible. But yeah, for live imaging, it requires a very different way of thinking.
Okay, we have a lot of interest in this this movie treatment for Alzheimer's, and
Yeah,
I believe it's Annabelle Singer.
that's right.
I guess we should probably, if you have any other critical references, several audience members are wondering, where can they go for more information on the that movie-based Alzheimer's application, particularly.
Sure. Yeah. Well, I have two links here, one from a journal and one from medRxiv. I don't know how to put links into the Q&A. Is there a way for me to respond to the Q&A? Or I see that I could type an answer. I will try to, I just put one link as a typed answer. Can you all see it now? And then I'll put the other link as a typed answer. Although now that I answered it, it's gone. But
and I think that if you
Catherine
can send it out.
The question was asked.
Yeah, we can see it. We can see
great.
it.
And great, because people replied back, I can have another link to reply. So I put Annabelle's paper is one reply, and then I put the paper that Li Wei led as a second one.
I can send this in a follow-up email as well to the attendees.
And then Li Wei and I have co-founded a company called Therapeutics, which also in abstract form has presented some interesting data. So yeah, those are the beginnings of the human trials out there.
Okay, another question sort of at a micro scale. Do you have any applications that you or anyone else is working on for radiation necrosis or remapping around pathways that have died off?
Could be, yeah. I mean, I'm an inventor, not an expert in specific diseases. So the way that we work is we try to collaborate with experts. I mean, Li Wei is an expert in Alzheimer's. They did the optogenetics to make the discovery of the brainwave. Then there was the design aspect. How do you make it non-invasive? Yeah, we've sent tools to thousands of people. So it could be, yeah. But the trick is I need to find like a biologist or a doctor to work with. And then we can help them use the tools towards it. But there's just too many problems to work on. And so that's why a lot of our projects, like the early cancer detection, which is done with a bunch of Harvard pathologists, or the Alzheimer's stuff, which is done with Li Wei's group, we look for an expert in that area, and then we try to help with our technology to see or control those pathways.
Somewhat related, just following up on that, do any of these tools help established treatment response. Perhaps for, I suppose, cancer, if there's maybe anything that would give doctors more insight into choosing the best course of action with a better, micro scale informed sort of basis for it.
Absolutely. I mean, that's kind of what we want to do, is to show people what's actually there in all of its detail and all of its glory. And yeah, so as I mentioned, with the Harvard groups, we showed that we could detect breast cancer at an earlier stage. Doctors at that stage actually disagree about half the time on the diagnosis. So it's kind of a major crisis. But now, we showed that we could train a machine learning algorithm on expanded samples to be far more consistent than when people are trying to squint at the tiny unexpanded samples. So why can't that be done over time to look at treatment response? I think it's an obvious downstream application that would be fantastic to pursue. But again, I would need to find a doctor who works on such things to work with, because, again, I'm not an MD. I'm good at inventing and helping people with technology, but I'm not an expert in any specific disease.
I guess that's another good question for you, Ed. There's just so many different possible applications that are emerging in the Q&A. Maybe you could just comment on, like, what are you most excited about? What do you think is closest to making its way into mainstream medical practice? Or is there a new breakthrough that you're working on at more of a basic science level that's really occupying your mind at the moment?
Yeah, maybe three thoughts. I alluded to all three briefly in the talk, but maybe I can elaborate on them. The first is early disease diagnosis. Again, something like 7 out of the 10 leading causes of death, if you can catch them earlier, you can save more lives. And I'm obsessed with this idea that early in a disease, the changes are just too small to be seen. If we can make them big and obvious, helping address 7 out of the 10 leading causes of death would be huge. No pun intended. Yeah, so I'm talking to lots of doctors about everything from glioblastoma to diabetes to everything in between. With the optogenetics, I think this whole idea of discovering brainwaves that can heal is very exciting. So what Liwei discovered for Alzheimer's, could that be done for other brain diseases as well? And then finally, I'm really excited about whether we can have theories of the brain. Every brain clinical trial is so much work and so much risk and they can fail in stage 3 for the most mundane of reasons. Why can't we be like more mature sciences like computer science, where somebody can drop out of college and start Microsoft or Facebook or something, and there's still a lot of risk, don't get me wrong, but it's not science risk, And so that's one reason why at the end I was really talking about integrating the tools and seeing if we can make models of the brain and so forth. It'd be great if we had a roadmap that would allow us to generally tackle all sorts of problems of the brain. That's a longer term vision, but that's the basic science arm of our group, basically.
Yeah, that seems like a really exciting educational possibility, maybe a kind of a longer range career goal. All right, looks like we maybe have time for one more question. There's one regarding a supramolecular complex. What happens if you expand that? A cell signaling complex in a membrane, for instance. What goes on with that being expanded, and what about metal dependent events, since metals are diluted? Sort of a variety of technical questions there.
Yeah. Well, the expansion of the proteins away from each other seems to be pretty even. I know, as I mentioned, we even could expand amyloid plaques, one of the toughest beasts in biology. So I'm pretty sure we're pulling the proteins and other building blocks away from each other. We've only validated down to 20 nanometer resolution, so not quite single protein, but we'll be there hopefully by the end of this year or next year or some other time not so long from now. As far as metal dependent events, so when we form the polymer, we anchor all the biomolecules in place, right where they are. So if the metals are washed out and now 2 proteins are suddenly changing, well, they're still anchored in place to the polymer. So I guess it kind of depends what the metal dependent event is. If it's about the proteins moving or whatever, well, the proteins are locked in place. But if it's about some other metal-dependent event, then it kind of depends on the details, I guess.
Okay, fantastic stuff. We've we've run the gamut on this question session from the micro level all the way to sort of broad medical applications, which seems metaphorically appropriate given your career trajectory and the focus on expanding the very small. So with that, we will go ahead and wrap up. If you've stayed this long, that if you can just stay on a little bit longer, we would like to say a little bit about the Brain Health Project. Lifelong brain health is our shared goal. And as you got a sense from this talk, the Brain Health Project is a large-scale landmark study which will help people everywhere get proactive about maintaining and improving their brain's health and performance. If you're interested in learning more about the Brain Health Project or possibly participating, there is a QR code which you can scan on the screen right now, or go ahead and visit us at thebrainhealthproject.org. That's thebrainhealthproject.org. So if you're interested in continuing education credits, please visit centerforbrainhealth.org/CEU, centerforbrainhealth.org/CEU. And thank all of you so much for joining us. And thanks so much to Ed Boyden for sharing his remarkable work. And we look forward to the next talk from Ed, when he will no doubt have some other new invention and an update on all these fascinating directions that he's moving in. Thank you.
Have a great day.
We work, we live, we innovate and create. At the center of it all is your brain health. The ability to solve problems, think analytically, share empathy, and thrive.
We're trying to make brain performance really the next fitness revolution.
So how do you boost brain power? Welcome to the Brain Health Project, an urgent call to transform your mind to work stronger and faster.
This is an absolute crisis, as great as any we have ever faced. We have to equip the minds and brains of our citizens to cope with the accelerating, dizzying rate of change that they face in their lives.
Your brain health is not fixed. Scientific discoveries prove it can adapt and grow regardless of your starting point.
Our greatest value, the asset that will help us to change everything, every problem we're in, is all in our head.
To harness that treasure, we must measure and monitor progress while things are going well, versus waiting for an injury or disease to strike.
Too many of us are outliving our brains, and that does not have to be the case.
The information age is bombarding us with more content than our human brains can handle. How do you keep from getting lost in this? And focus on deep thinking. For starters, stop multitasking.
Science shows us that multitasking is bad for your brain. It reduces fluid intelligence, causes brain atrophy, and increases chronic stress.
The global pandemic is creating more stress than ever, stress that leads to depression and anxiety and beyond. Unlocking our potential to navigate these hurdles starts with learning the right strategies, even in school.
So when teachers have these strategies, they're empowered to support our learners, and then the learners are now able to take ownership of their learning.
Training kids how to think is doubling academic achievement among middle schoolers.
I think the greatest national security threat is pre-K through 12. If we don't take care of educating our young men and women, then we have to ask ourselves, where are we going to be in 20 years?
Our world-renowned scientists know you can increase your brain health, not lose it. It's time for a new category of health. Brain health. You are a game changer. Ready to transform the world with us? Be a part of the brain health revolution. It starts